Okay, now let's see what happens if we look at protein folding and how hydrophobic and hydrophilic interactions can affect that. So this simulation is showing you a polypeptide. So this is a polymer of amino acids. So here, each little like sphere represents a different amino acid. And the amino acids with hydrophilic R groups, so polar R groups, are coded green. And the amino acids with hydrophobic R groups, so nonpolar R groups, are coded yellow. So this protein or this polypeptide, let's put it in some water and we'll press play and we'll see how just the random movements of the amino acids will kind of make the protein fold into a certain shape. Let's see. Okay, so do you notice a pattern? So see, all of these hydrophobic amino acids have clumped together, right? And they've clumped together in the center because they are afraid of water. They are hydrophobic, so they're trying to get away from the water. Whereas on the other hand, the hydrophilic amino acids are all on the outside for the most part because they like water. And so they're attracted to the water molecules that are surrounding this polypeptide. So that's if our polypeptide is in a water environment. We can do the same thing if we place it in an oil environment. Um, Oil seems a little bit silly, but this really does happen in your cells. Not in oil, of course, but you do have proteins that are inside of cell membranes, which are lipids. So this is an accurate depiction. So I want you to just think for a second, what do you think is going to happen when I run the simulation now that it's been in oil? Let's see. Here we go. It's the exact same protein. It goes, oh, oh, okay. There we go. It was kind of stuck. But yeah, so you can see the protein is essentially starting to turn inside out. So now these hydrophobic amino acids are starting to come to the outside, and eventually the hydrophilic amino acids will go all the way to the inside if we give this enough time to totally reset. Um, this is actually even more drastic. If we show you, let me show you a protein that is, there we go, let's do mostly hydrophobic. And let's put this in water. So if we take this protein that's mostly hydrophobic and we put it in water, all the hydrophobic amino acids are going to clump together, right? Like a big old clump of oil. So now if we take the same protein and we put it in oil, oof, there you go. Now you can really see it essentially flips inside out. And now the hydrophilic amino acids will be on the outside and hydrophobic amino acids will be on the outside.